Have you ever wondered how much DNA is in each of your cells? Each cell has an entire copy of your DNA, even if it doesn't use it all. If we stretched out the DNA in just one of your cells, bearing in mind that you can't see a cell with your naked eye, it will be about 2 meters long. If we then took all of the DNA from all of your cells and lined it up all together, you would be able to wrap it twice around the diameter of the solar system. We can see then that packaging of the DNA is very important if we're going to be able to fit it into a space just 6 microns in diameter. And for those of you who don't know what a micron is, the units go meter, centimeter, millimeter, micrometer. And there are 1,000 micrometers in 1 millimeter. So your 2 meters of DNA all have to be packaged into 46 chromosomes, 23 pairs. Histones are a protein and they're positively charged, and they can be used to wrap up your negatively charged DNA. And you just keep wrapping and wrapping until you end up with a chromosome. You'd be surprised at how many people think a chromosome looks like an X, but it's actually an easy mistake to make. The X shows you a pair of chromosomes. One chromosome is just one side of the X. And all of these chromosomes have to be held within the nucleus. The nucleus is an organelle present in almost every single eukaryotic cell, with the exception of a few specialized cells such as the red blood cells. And for those of you who may not know, a eukaryote includes plants and animals, not bacteria. As you can see, the nucleus is a spherical organelle, but what you can't see is that it's surrounded by a membrane called a nuclear envelope. This envelope is perforated with small holes known as the nuclear pores. The function of the nucleus is to hold all of the genetic material belonging to that organism, the DNA. The function of this nuclear envelope, or the nuclear membrane, is to separate the genetic material from the cytoplasm of the cell, and it also blocks other molecules from interacting with the DNA, which could possibly be harmful to the organism. However, when genes need to be translated into proteins to maintain the cell and its surroundings, copies of the gene have to be able to leave the nucleus and interact with the ribosomes in the cytoplasm. The nuclear pores regulate this movement. Because the pores are only so big, it means that it blocks the large molecules and organelles from getting into the nucleus, but allows smaller molecules such as RNA to pass through them. Within the nucleus is the nucleolus, which is not surrounded by a membrane. The nucleolus synthesizes ribosomes, which are a key component of gene expression. As I mentioned before, plants and animals are known as eukaryotes. Bacteria, on the other hand, are known as prokaryotes, and they do not have a nucleus. They still have DNA, but they just don't hold it in a contained and membrane-bound nucleus. Instead of being linear in eukaryotes, prokaryotes have circular DNA which rests in the cytoplasm. In addition to this circular chromosome, bacteria also have additional lengths of DNA known as plasmids, which may contain genes of high importance, such as those that give cell antibiotic resistance. Again, these are circular lengths of DNA and they can replicate separately from the chromosomal DNA. Thanks for watching, guys. Please subscribe and comment down below with anything else you'd like me to teach you. Bye!